In question 72, we're given the following table showing DNA-based triplet codes that are valid for this question and the amino acids that they code for, and then we're given this DNA sequence, CGC, AGT, and obviously this is part of a broader sequence. We're then told that two mutations will take place in the DNA sequence. The initial mutation will be the insertion of an additional base between the first and second bases in the sequence, i.e. between C and G here. The second mutation, which will come after the first mutation, is a deletion that removes one base from the sequence. Okay, bearing that in mind, assuming that this DNA is part of a longer sequence coding for a protein and that no other mutations are occurring, we're asked the probabilities of two individual occurrences. Firstly, the probability that after the first mutation, i.e. the mutation of insertion between bases 1 and 2, the first triplet does not code for arginine. Okay, well, the second probability we have to think about is the probability that after the second mutation, i.e. the deletion from one base from the entire sequence rather, both triplets code for arginine. Well, let's start with the first one. We know we have our sequence here, and having a look at our table, CGC codes for arginine, whereas AGT codes for serine. So at the moment, the first triplet does code for arginine, but we will have a insertion mutation between bases 1 and 2. Now, what sort of insertion could possibly leave arginine as a potential outcome? Well, the only possible mutation would be an insertion of guanine between C and G, because the resulting triplet will be CGG, which is still arginine, as we can see here. So, in order to prevent this from happening, or rather we should say the probability of this occurring is 1 in 4, since this could be A, T, C or G, and the probability of this not leading to arginine for the triplet, i.e. the probability that the mutation is not guanine, is 3 in 4. So, in order to work out what the probability is that the triplet does not code for arginine, we have to first think about what could possibly lead to arginine occurring and working out what 1 minus that probability is. Okay, so we have our answer. G is the only potential mutation that could lead to arginine persisting. We can bear that in mind coming into this part of the question, which asks for the probability that after our second mutation, just as a reminder, this is the deletion mutation from one base within the entire sequence, both triplets code for arginine. Okay, well, we already know that our insertion of G is the only way that we can continue having arginine for the first triplet, so we can bear that in mind. And secondly, in order to have the second triplet, i.e. the CAG, continuing to be arginine, or going from being glutamine to being arginine, we can get rid of the A, and that will lead to CGT, which, as we can see here, codes for arginine. And this is the only deletion that we can do that will lead to a second triplet coding for arginine. So, in order to work out this probability in total, we have 1 in 4, which is our first mutation being guanine, multiplied by 1 in 7, i.e. this 1 within the 7 long sequence being the one that's chosen for deletion. So the total probability is 1 in 28, and 3 in 4 and 1 in 28 gives us our answer E, row E within the table. So we have our answer.